those of you who have come along today to hear all about biosecurity strategies in the Australian beef industry, you might be a little bit disappointed. So what I'm going to talk about today is a little bit about actual stuff that goes on, a little bit of virology, and for those of you who know me, I'm a virologist by trade. But then what I also want to talk about is some futuristic type things that might happen in the future. And I'm also the seminar coordinator for QAAFI this year, and I hope that some of the concepts that I introduce towards the end of my talk, that we'll have some more informed speakers later on in this seminar series throughout 2015. So excuse me. So I'm going to start by outlining what biosecurity is, and that's a very nefarious term. If you go onto the internet and put in biosecurity, you're going to get a whole variety of different things. And so I'll just sort of set the scene of what the context that we're talking about it here today, and that is within essentially livestock industry within Australia. Talk a little bit about how it's currently managed, and that's mainly a little bit of politics and a few other things like that. And then go into where we might be in the future. That might be 10 years, 20 years, or perhaps even further. So what is biosecurity? For the purposes of today, it's about maintaining the disease status. So it's keeping things in, keeping things out, if we know that they're there. It's about monitoring, and if we detect something, we report that. So it's about finding the one that stands out from the crowd and what's unique about that animal with our respective disease. And then for most of the current biosecurity strategies, it's about managing that situation as it evolves. Now, those of you might have seen, so this is from the transfer of the Ebola patient from Texas, I think, to CDC. And in biosecurity, everybody has to play their role. So we have the patient here in yellow, easily identifiable. We have all our guys in biohazard suits. And then there's the clipboard dude. Now, what he's doing, who knows? And in subsequent shots to this, he actually does go up the walkway into the plane in the area. So you can make up your own mind about that one. So for biosecurity in Australia, it's a heavily regulated area. So we trade on our freedom from those diseases that we test for, and those diseases that we know that we have, and particularly those things that we want to keep out that underpin our market shares. So the federal government coordinates that through Animal Health Australia. There's a whole, I won't call it a rigmarole, there's a whole system in place across the country whereby the federal governments and the federal government and the state governments all each play their own particular role in biosecurity. That in some ways is underpinned by our National Livestock Identification Scheme, whereby any cow, once it leaves its property of origin, we can trace that back to that property of origin if anything goes wrong with that particular animal. And so that stemmed from the BSE cases in the UK all those years ago. So as I said, the feds coordinate it, the states try to implement it. So there's a whole series of plans in place. If this happens, this is what's going to happen, and who pays the bills at the end of the day. And so the states are responsible for most of that implementation. The front line in this scenario is our veterinarians and our producers. So those people that have the most contact with those animals on a day-to-day basis, they're the ones most likely to see something if it goes wrong. Is it important? Sitting here on our relatively big island, yes it is, because we trade on that freedom from disease status that we have in world markets. The best example about that, and I'll talk a fair bit about foot and mouth disease today, so that's sort of the big bogeyman, particularly with respect to beef, if that was ever to come into the country. So there's been a lot of work done, that again in Canberra, about what would happen if we got FMD into the country. Quite in one word, disaster probably was what it would be. Modelling for a large-scale multi-state outbreak, so if it went all across the country, estimated it would cost us about $60 billion over 10 years. And depending on if we use vaccination as part of that control process, and I'll again go back to that again, I have to declare a conflict of interest there, is that my group mainly focuses on making viral vaccines. And then once we were to regain our FMD-free status, we then have to get back into those markets that were lost. And that might be the biggest challenge of it all in the current competitive market. So, as I said, it would be massive on the beef industry in Australia if we were to get FMD here, particularly in those sectors which are export driven. So that's where most of the money comes from. They would be shut down immediately, and we'd have cows everywhere. Interestingly, when they do model this, they consider that for us domestically it could be a benefit, in that the domestic markets will be flooded by all this beef because they can't export it. So for us on the ground, we might actually have reduced profit or reduced cost of beef. The question is unknown there, I think, is about whether, even though people know that it doesn't infect humans, whether it would actually be acceptable and whether they would actually eat it is another question. So where are the cattle? Distributed mainly, as most of our industries, around the coast, a little few over here, around here, and mainly in Queensland. So we have half the national herd here, so it's a big issue for us. And then we have these, as we sort of go towards the middle from either side and up and down, the density of cattle reduces, of course, due to the quality of the land. And therefore you might think, well, that's a pretty good strategy. If we get FMD down here, then if we can cut it off here, then we should be pretty right. We'll keep it out of those big northern producing areas. That is until you overlay those things that we can't control. So in terms of feral animals, so we have a huge problem with feral animals. A lot of those are susceptible to FMD. We can't control them now, so how would we control that in their role in an outbreak like FMD? So a few things to consider there. And that comes back to our primary way we implement biosecurity now, and it's largely a passive process. So we have a list of diseases we're concerned about. Top of that list is probably FMD for cattle. And then if that happens, we have a list of measures that we will implement when that occurs, once we know about it. We collect samples, we test for it, and a great example of that is the great work that went on, I can't remember what it was now, with the equine influenza, again a few years back, whereby the state labs were testing thousands and thousands of samples during that standstill. And that's the first step, is no one goes anywhere once it's identified. And that creates a whole bunch of issues in itself. And so we're being passive and responsive in that process, that we try to manage it once it's here. And I'll come back to that as a central sort of theme. So we have this process where we continually surveil animals and we try and look for things, and you've got to get the right animal. Critical to that, as I said before, is if we were to have a large-scale disease, be it FMB or something else, if we had a vaccine available, then we really have to consider that we would use it. So the critical thing with FMD is if you vaccinate those animals, then if you can't tell the difference between them and naturally infected animals, then everybody goes into the pit at the end of the day. And so even though you might stop the outbreak, you do expand that economic impact. And as we move to be more reactive to proactive from reactivity, the problem is that we get things from industry, well, we can't really do that. But I think we all accept that we're actually changing the way, or animals industry will have to change with the way the public perceives and consumer acceptance in the future. An example of that is I've got here on your bottom right-hand side is the way the feedlot industry manages heat stress. And so when you look at it from the outside, heat stress is caused by climatic conditions. The general concept is, well, if it's climate or if it's weather, well, we can't do anything about the weather. But with heat stress, it's all about being aware that based on type of cattle you have, the weather conditions at that particular time, and how they're forecast into the future, you can actually predict with reasonable accuracy what's going to happen to you in your feedlot in the next week or two. And a great example of that is that last week Alpha was putting out, the Australian Lot Feed Association putting out warnings about potential heat load issues in these coming weeks. So if we really think about it and not just about being able to do things on the ground, then there are things we can actually do. So, as I said before, my conflict of interest is that we develop vaccines in my group. So we don't develop FMD vaccines, so I use that as the example. So in the UK, it's about 14 years ago now, there was an 11-month outbreak. It cost them somewhere in the order of $10 billion US dollars, depending on how you calculate it. Here is an outbreak in California in the early 1900s. So for all younger people, that's, it was like last century, the beginning of last century. And you can see there that not a real lot's changed in that 100 years between the managing of those outbreaks. So I'm not sure what Libby, our OH&S person, would make about this, people in the pit and then shooting the cattle. But if you have OH&S issues there, we might deal with those a little bit better. But by and large, as I said before, everybody ends up in the pit at the end of the day. So there was a lot of debate around this, not using vaccination because, as I said before, at the end of the day, if you can't tell the difference between vaccinated and unvaccinated, then everybody goes. Because the greater cost of this outbreak was, this one was not so much, was agriculture certainly, and the standstill that ensued, but also with the closing down of the, what do they call them, the ramblers in the UK. So all the paddocks and everything, everyone likes to wander about the beautiful English countryside. No one was allowed to do that. So there was huge losses in tourism industry, and they were the ones pressuring the government to try and get them to do something about this more quickly. But here is the crux of the situation. There was a little over 2,000 animals were confirmed to be infected with FMD in this outbreak. There was over 10 million animals culled to get back to that FMD-free status. Now we know the pressure that our industries are coming under in terms of the animal welfare lobby, and so you can see, or hopefully you can see, that does create a dilemma in that you're killing these otherwise healthy animals to get back to this disease-free status. But apart from that ethical dilemma, it's also a logistical dilemma in that you need to find somewhere to put all those animals and do something with them. And similar to that, there was an outbreak, I think it might have been associated with this one, in the Netherlands, where they only had 26 cases, but again, with the slash and burn policy, there was, was it 100 times, 1,000 times that number of the actual cases that they had was slaughtered to try and get back to that disease-free status. So I think we probably would all accept, particularly with some of the events that have happened in Australia in recent years, that relying or being totally reliant on this sort of policy is going to be changing in the future, whether we like it or not. The other side to that, there was an effective FMD vaccine available at that time. But as I said before, the difficulty was that once you've started to use vaccination, it makes it more difficult for you to track where your actual cases are. And so then when you draw that line in the sand and say, okay, we're right to go forward, you then have to make the decision about those vaccinated animals once your outbreak's controlled. And the key thing there is, and I'll talk a little bit about that in the next few slides, is that being able to differentiate. And so there was an outbreak of FMD in South Korea. They decided to vaccinate, but then 5 or 6 years after they controlled that FMD outbreak, they were still an FMD country, even though they hadn't had a case for a number of years, because they had positive animals. So what do we do about that? So there's this concept going on in vaccination, and that is around what we call DIVA, or differentiation of infected versus vaccinated animals. So our normal test when we look at if an animal's been infected with a virus or something, we would test for antibodies. So you can see here we've infected with virus, make it your favourite virus, and the animal has raised antibodies to 2 of the antigens, or 2 of the components of that virus, and so we test for those and then we know it's positive. So what people have been doing, both with respect to FMD and some other viruses, what you can do is you can take out one of those antigens. So obviously it's not something that is critical for protection against infection, but it's part of that immunological response to vaccination. And so then when you test this animal here, we can say, okay, this one was vaccinated because it doesn't have antibodies to this. So that's all very well until you work with some of the viruses that we work with, and then you can actually find naturally occurring mutants that don't have that particular antigen. So you do have that, there is a problem there with terms of positive, when you're positive as a negative, can make it difficult to interpret. So what we have done with some of our vaccines is we've introduced an antigen from something that we would try to predict cattle would never be exposed to. And so therefore, if we see an immunological response to that, then we know that that animal has been vaccinated. And the gene that we've used nominally in my lab is green fluorescent protein, because generally speaking, jellyfish don't attack cattle, usually. So the process that we use for that, we were one of the first in the world to start using this process. So normally what we would do is we grow our viruses in cattle cells, and then we would use homologous recombination to introduce changes to that virus, whether it's putting genes in or knocking genes out. And then we take those back and we vaccinate our cows. So what we have developed in our lab is a process whereby we introduce those viruses into bacteria. And once we have them in there, we have a much greater capacity to actually manipulate the genomes of those viruses, and we can make those changes, be it gene deletions, gene insertions, antigen genes or reporter genes, all sorts of things. And then once we've made our changes and we can fully characterise that, so it's a lot easier to make those changes in bugs, we introduce them back into cells, and then we have our recombinant. So here we've inserted our green antigen, nominally GFP, and then we can go ahead and test those in cattle. So we were actually supposed to start an experiment like that yesterday, but then all our cows got infected with something else. So now we have to wait for another month or two till they get better, so we can make them sick again. That's life with animals. So it allows us not only to develop these DIVA vaccines where we can differentiate vaccinated from infected animals, but we can put in genes from other viruses, so we can vaccinate with one virus and then they become immune to two viruses. And that's what my group's been focused on for a number of years now. So using these sorts of approaches, and some stuff I'll talk about now, is about moving to a proactive biosecurity arrangement, whereby we don't wait for something to happen, we don't wait to detect that index case or have to trace back to find that index case 6 months later, so that we can better manage and better contain the situation. So as I said before, we usually have to find that animal and then things start, if we do find it. And so it's always catch up. And so once the cat's out of the bag, it can be very difficult to put the cat back in the bag or the cage. It's usually identified by some unusual clinical signs. So that's the animal's been infected. It's gone through that incubation period whereby, you're feeling crook and then you get sick. Animals don't usually complain that much. So we have to wait for those overt clinical signs. So again, it delays that response, inactivation of that response. And then we have this situation whereby we say, okay, we've got this animal that's got lesions on its mouth and there's something wrong with its feet. Is it FMD? We take some samples, we send them to the lab, they do some testing, and if it's FMD, they're very likely to detect it because we have a great system in place for FMD in Australia. But all of our existing tests are largely, we have to know what we're testing for. And so as we've seen in the world over the past couple of decades, there's new things coming out all the time in response to our behaviour in the planet. And so it's becoming, or can be quite difficult to detect new pathogens that aren't related to things we already work on. So with the advent of new technologies, and here I'm talking about next-gen sequencing, those sorts of things, if we can better understand the difference between an infected and an uninfected animal and pull out those central themes that allow us to say, okay, that's got a virus. We don't really know what it is, but because we don't know what it is, that's something we really need to manage proactively. And so one of the things we've become interested in the last few years is biomarkers of infection. So rather than detecting the pathogen, it's just about saying there's something different about that animal. And if that's generic across a range of different pathogens, then it makes it much easier to identify it. And you don't need to know what the pathogen is. And while the whole focus of my talk and when we talk about biosecurity, by and large, we're talking about endemic exotic agents, so things that we don't have in the country, but we don't sort of worry too much about endemic species, pathogens. There's a whole range of subclinical infections out there that probably reduce our overall productivity. So if we're better able to understand the disease development process, then we might be able to manage not just exotic diseases better, but also those endemic pathogens in our animal populations. So an example of that I'll give you is there's a small German town called Schmellenberg, and as you can see, it's quite picturesque. And so, just like Hender in Brisbane, it now has the envy of the world in that there's now a new virus named after it. And so that emerged in the cattle population in Europe about 4 or 5 years ago. The animals were sick. They were able to isolate the virus. They could actually grow it in the lab. They knew they had something, but it was negative for every test that they could put across that virus. So in the end, what they did was they applied next-generation sequencing to that, so just generic RNA sequencing, and then took the results of that and did some broad screening blast searching, and they identified a relationship with a very distant relationship with a known virus called Akabani virus, which is actually an Australian virus. And so without the advent of this technology, they had no idea at all. So it was negative in every test that we had. So an example whereby we can identify new things of importance. We take that back to the other side and then talk about the host. And as I said before, about identifying markers of disease. Now, we've done this historically when we talk about doing serology. So we'll test an animal for antibodies to a particular virus or a pathogen that we're interested in. And then we know that sometime in the distant past or recent past, that animal was infected with our agent of interest. But what I'm talking about here is actually taking that part of it away and then being able to test for what's the status of that animal now. Is it currently infected with an agent that we know or unknown? Or is it healthy? And so we find that those two things very difficult to differentiate. So once an animal starts showing signs, that's not a problem. We know we have to do something. But what about in that intervening period? Would we be better off taking that animal and putting it somewhere else? And so there's factors that will be specific to the host, so the host response to infection. There'll be factors that are specific to the pathogen, so how the pathogen goes about that infectious process. And so what we're in some ways talking about there is what we call virulence, so the capacity of an agent to cause disease. And there's many things that we know about that cause disease. By and large, we don't know a hell of a lot about how viruses and bacteria and a lot of other pathogens actually cause disease. So how do they enter a cell and take over that cell with such exquisite control that we would love to be able to replicate ourselves? We can make attenuated ones, so we can modify them sometimes specifically, sometimes non-specifically, and that's where a lot of our vaccines have come from. But again, we don't really, in a lot of cases, understand why that attenuated virus is different to our one that causes disease. We do know a little bit about antigenic shift and drift. And so in the context of, say, influenza, that those of you who get the vaccine every year, we see changes in the strains of circulating influenza in response to the vaccines that we use each year, and we would call that process antigenic drift. So they change slightly so that our body can't recognise that new virus. We then have situations which we refer to as antigenic shift, when not the whole virus, but large parts of the virus changes in a fairly short period of time. An example of that would be the H1N1 that emerged a few years back, whereby they acquired a different antigen gene from a different virus, and so therefore nobody has any immunity to it. So we have all those sorts of scenarios. So about understanding what pathogens do when they enter animals. So I'll go into couple of more specific details now about this, how we've been looking at this process over the last few years. So Gallard herpes virus 1, or Marek's disease virus, is a large DNA virus, so it's a herpes virus, closely related to those viruses that cause cold sores in humans, except in chickens it causes a T-cell lymphoma, i.e. cancer, and causes high mortality in chickens. For the last 40 or 50 years, Marek's has been well-controlled disease, i.e. the expression of tumours, has been well-controlled through vaccination, again through largely unknown mechanisms. And it only, as I said, it only prevents tumour formation. It doesn't prevent infection of chickens, it doesn't prevent replication of the virus in chickens, and it doesn't prevent shedding of the virus into the production environment. So whether you like it or not, you've probably consumed Marek's disease virus at some time. So this continuous cycle of vaccination infection means that our vaccines are continually being, lack of a better word, challenged with respect to how they provide that protection. As a consequence, this is what we call our vicious cycle. So if we don't vaccinate, we might get 60% mortality within some flocks. If we do vaccinate, the virus can still infect and goes back through subsequent flocks, so that's continuous probing of the protective effects of vaccination. And this is a model that was developed in the US by Bob Witter. And as you can see there, they started vaccinating in the early 70s with another virus, but because we didn't prevent infection or shedding or replication, not in that order though, we saw the emergence of new virulent viruses that were able to overcome the protective effects of those vaccines. And this has happened a couple of times, and the viruses, so we call them, that was the Marek's disease first isolated by Joseph Marek. We then went to a virulent form of this virus around the intensification of chickens. We then went to a very virulent, and then in the early 2000s we were up to a very virulent plus. So that means it was able to overcome the protective effects of our best vaccines. When you take all those viruses, so the different phenotypes of these viruses, and sequence them, there's very little difference between them. So what we're hoping there is we identify this particular gene or this particular element that says, okay, this is why they can do this, but we weren't able to do that. So there have been things identified, but when you look at a large range of viruses or look at them in chickens, they don't hold up and don't make any sense. So, said it's not minimal sequence divergent, it's not antigenic shift as I described with influenza, it's not antigenic drift either. So what's the mechanism of that? So there was a study published, 7 years ago now, by a group in the States, and what they showed was that these class of non-coding RNAs, called Micrornas, are differentially expressed and related back to the virulence of those viruses. So you can see here, so these Marek's disease viruses express a lot of microRNAs, and as you can see, you work your way across there, that these are largely the same, regardless of the virulence capacity of the parental virus. But then we have these ones here, that as we go up into the higher virulence viruses, we actually see an increase in expression of those particular microRNAs. And so this provides us with a way that we would actually be able to potentially predict the virulence of those viruses, which we haven't been able to do before. Now, for those of you who don't know, quick lesson into non-coding RNAs, and I say because it didn't exist when I went to university. It didn't exist when I graduated from university either. So it's a real paradigm shift in how we thought about genetics, when it used to be just we had a genome, we got a RNA, and then we got a protein. So channeling John Maddock here from the 6th tower, the IMB tower when he was here, about the role of these non-coding RNAs that we used to consider junk DNA within our genomes, but really are playing some significant roles. So they allow us to explain the complexity between animals that have similar numbers of genes, but overall are different organisms. They provide the capacity to finely tune biological pathways in relatively real time, and they play a big role in pathways that are required to do that, such as the immune system. And as I said before, what studies have shown in Marek's disease, that they're also linked to the pathogenicity of organisms, again, that we haven't been able to explain before. So one of the viruses that we work on, back to cows now, away from chickens, is bovine herpes virus 1. So a similar situation to Marek's disease. It's a major pathogen of cattle worldwide, not just in Australia, but it's an endemic virus here, as it is where there are cattle in the world. And it's involved in a variety of diseases related to the reproductive and respiratory systems. But again, with Marek's disease, we've had a similar situation whereby we have differences in the capacity of these viruses to cause disease, i.e. virulence. And so we've been trying and looking at a number of different avenues to try and explain this over the past 20 years. So we know that they're different, so we can see broad-scale differences in their genome. They're somewhat associated with different clinical signs in cattle. So the type 1.1 are mainly associated with respiratory tract infections. We then have, and abortion is probably the key point there. In Australia, we only have, allegedly have the 1.2b subtype, but the difference genetically from these, so about 135 kilobase genome, is less than probably 2%. And considering that we have quite severe respiratory tract infections in Australia as associated with BHV, it's unlikely that it's totally to do with clinical signs. So we showed a number of years ago now that BHV does actually encode microRNAs. So similar to the Marek's disease viruses and all the herpes viruses for that matter, so the same with one that causes cold sores in humans, we see a clustering of microRNAs in particular parts of the genome. And then also throughout that. But by and large, we don't know anything at all about what these microRNAs are doing. There's been a lot of work done with the Marek's disease ones because there is that link back to cancer. But with respect to cattle, very little work at all. In fact, I think we're the only ones. So this is just an example of what it looks like when we do northern blots for these particular microRNAs. And so there's a student just started in my lab, and so Trisha's actually going to look at if we can use these molecules to tell the difference between strains of BHV, which we know behave differently in cattle. In terms of the host, so can we use microRNAs as biomarkers for infection in cattle? So again, as I said, there's been very little work done in cattle. This is an example of people, so humans, infected and uninfected with tuberculosis. And as you can see there, I've cut this down a bit, we have one microRNA here, and I just love these scatter plots. Can you think, are they really different? But we have to go with the authors on this. So we see differential expressions, so some are clearer than others. Maybe that one, definitely that one. So there we're seeing that the host, or these microRNAs, are behaving differently in uninfected and infected animals. And so there are particular microRNAs, again, if we had a lot more time, we could go into and the roles that they play. And we don't, in most cases, we don't know a lot about what these microRNAs are doing in the cells, what genes they're regulating or what genes they're affecting. As I said, in the bovine space there's been some activity in terms of some production traits and some work in mastitis as well. So we require a lot more work to be done in cattle. There's a number of microRNAs that are being increasingly used as biomarkers for different things in humans, particularly in cancers. So they actually can detect those in sera. And I guess where I'm heading with all this is that I think that in the future, if we're able to do enough work on these, that we should be able to identify the infectious status of animals independently of what the agent actually is. The other advantage of this particular class of biomolecules is there's also that potential there that we'll be able to say what's the virulence potential of that organism if we know about it. So if we can identify what the host is doing, we might be able to identify the type, the identity, and what's the true risk of that particular virus that's there. So what's the feasibility of all this? Currently, this is where we get to the really speculative part. So this is the bits, i.e. the bits that I've made up. So as I said, there's ongoing work in various systems, so we're not quite there yet in terms of what these molecules might be doing. But we know general signs, or even in humans, symptoms of disease. We have increases in temperature, we have increases in respiration, and for animals, and I guess some ways humans, we know that when they get sick, you tend to lie down and have a rest. So if we're able to track that, we would be able to see that in cattle potentially. There is a range of molecular-based detection systems that are being developed for human medicine. So again, things, for those of you old enough, to the Fantastic Voyage, where you shrink down a submarine and inject it into someone and they go off and do something. So this is a glucose monitor where you have a biosensor and then electronic stuff, and that all ends up in being a signal being produced and transmitted out of the body. And so there are systems that where companies are developing, as you have this coupled with a glucose pump or an insulin pump I think, and so therefore for diabetics being able to better regulate the glucose levels in their body. This is apparently a blood pressure monitor. So when you consider this is the ridges on someone's finger, that's pretty small. So I guess what I'm talking about here is we're really trying to bridge that gap between, once we know about our biological system, being able to develop systems whereby we can actually monitor those remotely. And so again, many of you probably know there's been a lot of work done in the sheep area. So with the sheep CRC, there have been e-sheep. So essentially trying to use remote systems to manage our livestock. Again, not so much in dairy herds, although there is a lot of work done there as well. But if you can actually monitor things remotely, so you don't need to go out, if you've got your cows spread all over 100,000 square kilometres, do you actually need to go out and see them every day? Obviously you can't. So there's one such system that's available called Cattle Watch, and you're essentially going to turn your whole herd into a Wi-Fi network. So I think my kids would be pretty excited about that. So you have what I guess is your hub cow. So that one might have a satellite receiver on it. And so these are actually real systems that are available. And there's a group talking at BFEX in Rockhampton in May about the system that they're looking to bring to Australia, or might actually be here. And so you have a percentage of your herd that has satellite collars, another percentage of your herd that has the collar units, so they might be reading. And these are not doing the sorts of things that I was talking about before in terms of in vivo molecular sensors, but they're able to monitor a range of different things. And then everybody has an ear tag, and so you can actually monitor where your cows are, what they're doing, and they're all connected to satellites. As you can see here, they've and I'm not really sure about how they determine these, but I imagine what you do is that you monitor a large enough number of animals over a particular time, and so you get to develop the degrees of accuracy in your system. And so they've classified all these different dots represent an individual animal and what's actually happening with that animal at that particular time. Seems to be a little bit too much going on in that herd at one particular time, but anyway. So, and then, farmer, I don't know what you call him, probably Billy Joe Bob by the looks, has his smartphone, and so he can monitor that when he's off going doing something else. And so we've talked about similar sorts of things in some other work I'm involved with, and there are various smartphone apps coming out to allow people to manage their cattle. So this cattle watch system, it provides you with a variety of different readouts for your animals and what they're doing at that particular moment in time. I think that they're mainly based on those sort of broad generic outputs rather than individual ones. And so I think that in the future, once these systems, and there are companies that we've been talking to that are looking at these sorts of things, then they'll also play a role in the management of outbreaks and epidemics. So in summary, I hope I haven't disappointed you too much with my talk today about actual biosecurity in the beef industry. It will continue to be a key component, and until we have the sorts of more outlandish systems that I've outlined later in my talk today to do that in our industries, then the more management style or reactive will continue to play a role. I think that we will be forced, whether we like it or not, to have increased adoption of vaccination in the control of outbreaks, and there certainly is. That's a component of how Australia would manage FMD if we were unfortunate enough to get it. But key to all of this is, if we are going to improve that situation and overall reduce that risk, is better understanding these systems. And so that's about understanding the biology of disease. So the one disadvantage that we have in animals is that you can make a vaccine and you can pretty much straight away go into what would be a phase 3 or phase 4 clinical trial in humans. And if it shows reasonable efficacy, then, and you can persuade a commercial partner, then that vaccine will end up on the market, maybe. The disadvantage of that is that we will then understand very little about how that vaccine works, and if it were to fail, then we don't talk about, well, maybe it was this, this and this. We go back to the drawing board and we generally start again. So if we can better understand these systems, then we should end up with better vaccines and hopefully better outcomes for our industries. If we have pathogen-independent strategies for identifying sick animals, then again we're not relying or being reliant on just looking for those things that we know about, particularly, I guess, with the number of diseases that have emerged in recent years. And when we combine that with the remote capacity, potentially, to be able to find those things without actually having to see those animals, then it will become extremely powerful. Despite we're not anywhere near this future yet, there are going to be new challenges for our industries as well. So we're talking about opening up the north to increased agriculture, including animals. And so as we move into those new environments, and then we potentially are faced with again, new disease issues. And in some cases, we're actually potentially moving our animals closer to risk than they currently are. We then have climate change, variation, whether you're a believer or not, on what the cause is. And so what the likely result of that is that we could see a change in the distribution of some disease vectors. So those areas where we haven't seen things before, they might actually move into that. And so there's a lot of work going on in that space about trying to predict where we might see changes of particularly the arboviruses that are spread by insects. as places get warmer and drier and wetter and those sorts of things, how that might change the scenario. Thank you very much for your attention.